Hello everyone, welcome to our lesson number 10, which is the last lesson of our module. Till lesson number 9 you have studied the different techniques, the methods, the concepts which are used in pharmaceutical analysis. Now in this last video you are going to study the real time applications of pharmaceutical analysis. You will also study the different case studies and you will also understand how pharmaceutical analysis is applied in the modern days. So not making further delay, let us move on to the topic. Lesson number 10, case studies and real world application. So dear students, this is our last chapter and today we are going to study the real world applications and the concepts which are applied. Now our lesson number 10 will explore the real world applications. We are understanding the real world applications of analytical techniques and which are used in pharmaceutical industry, and we will also study some cases. By the examples we will study how the things work. Also we will study how these techniques are applied to solve the practical challenges that we are encountering and how they contribute to the advancement in the pharmaceutical sciences. Now let us start the case study. We are going to understand case study by 3 examples which we will take one by one on the single slide. First is application of HPLC in quantifying API. Second is using mass spectroscopy to characterize metabolites. And third is implementing NMR for the structural elucidation of a novel compound. So case number 1, let us understand. Our example number 1 is the application of HPLC in quantifying APIs in a drug. Now let us take an example that you have formed a formulation. Say it is a tablet. Now this tablet is produced by a pharmaceutical company and this is a new tablet formulation and it contains multiple active pharmaceutical ingredient APIs. So let us assume that this tablet has API number 1, API number 2 and API number 3. There are 3 active pharmaceutical ingredients in one tablet. Now our challenge is that we want to accurately quantify each API. We want to ensure or we want to check what amount of A1, A2 and A3s are present in the sample. For example, let us say it is expected that A1 here should be 100 milligram, A2 should be 10 milligram and A3 should be 50 milligram. Let us assume that we have, while manufacturing, we have taken these quantities. Now when the tablet has been formed, we want to check whether a single tablet contains 100 mg of A1, 10 mg of A2 and 50 mg of A3 or not. So what we are doing is that we will check the quantity of each in the sample. So we will accurately quantify. The formulation obviously it is complex and we need to quantify A1, A2 and A3 because it will ensure the quality and it will obviously comply with the regulatory standards. So to solve this problem, what we are going to do is we will use an analytical techniques. In this problem, we will be using HPLC, high performance liquid chromatography. This high performance liquid chromatography will detect A1, A2 and A3 in the sample, and it will also identify as well as it will quantify the amount of A1, A2 and A3 in the particular sample. So by this way we can easily quantify the amount of A1, A2, A3 in the tablet. So this is how HPLC solved our problem. So what was the outcome? So HPLC enables precise quantification of each API. It has ensured that the tablet formulation meets the dosage specification. It means that one tablet contains the specified amount for real, and obviously that will comply with the regulatory requirements. Now example number 2, we are using mass spectrometry for characterizing metabolites in a drug metabolism study. So understand that scenario is like there is a preclinical drug development. We are developing a drug and we want to see what are the basic metabolites, what are the metabolites of these drugs. We want to know these metabolites, we want to identify them. So our challenge is to identify and characterize these metabolites, and we want to understand what is the metabolic fate of that body. That means when the drug goes into the body, in which processes it is undergoing for the metabolism. So to check, identify them, we have used mass spectrometry over here. What mass spectrometry will do? Mass spectrometry will check the metabolites. Say for example, for this drug we are having metabolite number 1, metabolite number 2 and metabolite number 3. Mass spectrometry will identify these metabolites through molecular masses, through detecting their molecular masses. It will detect the molecular mass of first metabolite, second metabolite and third metabolite, and by that way you can easily identify, you will get the info, you will get the information, and you will easily identify these metabolites and understand how the drug is going through metabolism, which metabolic processes it is undergoing. By getting the molecular mass of this compound, you will get the molecular composition of these metabolites. So what is the outcome? Outcome is that our mass spectrometry is revealing the structure of metabolites. It is helping us in the assessment of drugs safety and efficacy. How? It will tell us that the metabolite which we are getting are not harmful, and we are also understanding the metabolic pathways through which drug is undergoing. Now example number 3. Example number 3 is implementation of NMR spectroscopy in elucidating the structure of novel pharmaceutical compound. Now scenario in this case is like there is a research team who has synthesized a novel pharmaceutical compound, a drug that can be used for treating disease, and they also want to check its potential therapeutic application. That drug is having a potential therapeutic application, it is curing something. Now our challenge is to determine, we have formed the novel pharmaceutical compound, but we want to determine its precise structure. We want to confirm that the intended structure of the molecule is this. So we have to determine the precise molecular structure of the compound so that we can develop it further. Now for that we are using analytical technique like NMR. NMR is a nuclear magnetic resonance spectroscopy. We are employing it to elucidate the compound's structure. How? As I have told you, let us say that we are doing a PMR, proton magnetic resonance spectroscopy. It will tell us the number and types of proton present in that molecule, and to by getting the number and types of proton present in the molecule, you can assume, you can understand whether the structure is correct or not. So the outcome is that NMR spectroscopy provides a valuable insight into the connectivity of the atoms in the molecule. It will help us in the compound optimization, it will help us in the study of that compound. There is one more important thing to note, dear students, is that you must understand that only one technique is not employed. Like for example, here in this example we are characterizing the molecule, but there is not only one technique which we are applying. We are not just applying NMR, we will also apply mass. NMR will confirm the number of and types of proton, right? NMR will confirm the number and types of proton, H. Mass will confirm the molecular weight, and henceforth. So different techniques are applied, but as this is a case study, I'm just giving you an example to understand how mass is solving the problem of structural elucidation. Now, insights from the case studies. Now, what are the important insights that we are getting from these case studies. Now, the common theme of these all techniques is that accurate and reliable analytical techniques are very, very important because they help us to overcome the challenges in drug development. Each case study that we have done, it highlights the importance of selecting appropriate analytical method. We have to choose the appropriate analytical method because there are several methods that are available. But we must have the basic understanding of the subject so that we can apply the particular analytical methods to meet the specific requirements of the study. Now, what are the key takeaways from these case studies? The key takeaways are that the choice of analytical techniques will depend on the nature of the pharmaceutical analysis. It depends upon what kind of pharmaceutical analysis that we are undergoing. Either we are quantifying it, either we are identifying the metabolite, or we are performing the structural elucidation. And when you have successfully applied the analytical method, it will definitely contribute to the decision making, and it will definitely ensure the quality and the safety of our pharmaceutical product. Now, the real world applications of the pharmaceutical industry. So in real world, we are using pharmaceutical analysis to perform the quality control. This quality control will ensure the consistency and purity of our pharmaceutical product. We are also using in the formulation development, constantly analyzing the drugs are required in the formulation development because we have to optimize the drug formulation. We have to ensure that our drug formulations are efficient and they are also stable. So we are constantly checking them through pharmaceutical analysis. And pharmaceutical analysis is ultimately important to comply with the regulatory guidelines. We have to meet with the strict regulatory standards because if we if we are not meeting with the regulatory standards, we will not get approval for the manufacturing of these drugs. And even if we will manufacture it, it will be recalled. Fine will imposed on the pharmaceutical company if they do not meet the regulatory guidelines. Now, the analytical techniques are also used in the drug development also. Drug development means before it is marketed, the drug is being developed. It is a simple molecule which is being synthesized in the lab, and now we are optimizing this molecule so it can be taken as a drug. So that comes under drug development. We are also creating its formulation. So drug development is a very crucial step. Obviously, it is a starting step, and in this drug development, we are constantly doing pharmaceutical analysis. How? We are constantly screening and identifying the possible drug molecules, the potential drug candidates. Today, we are having high throughput screening, HTS technique, which is doing very fast screening of molecules. Now, we are also testing stability. We are constantly testing that our drug formulations that we are formed. See, all the drug molecule or one drug molecule cannot be formed in various different kind of formulations. It is possible that if you are able to make the tablet, maybe you are not able to make the syrup or parental of that drug molecule. You need to understand that. We have to check which drug formulation should be made, and we also need to check that in that drug formulation our compound is stable for its given shelf life. So stability testing is a part of analytical procedure that is constantly done to check the stability of our formulations. We also are interested in the impurity profiling of during the drug development, because impurity are the unavoidable part of formulation. It is said that impurity cannot be completely removed. You cannot remove it 100%, but they can be minimized. so that they can be taken safely. So we need to constantly profile the impurities. We have to check the impurities which are being formed in the formulations, or while we are manufacturing the drug molecules in the lab. So impurity consideration is very important, and for that consideration we are using pharmaceutical analysis. Now, what are the future trends and innovations in pharmaceutical analysis? So the future trends could be like that. There will be advanced analytical instruments. We are constantly developing these instruments which are of high resolutions. They are miniaturized instruments. Artificial intelligence is popular nowadays. It has also entered into pharmaceutical analysis. We are integrating this artificial intelligence. We are using it for the data analysis. We are also using it for the interpretation. Personalized medicine is one of the important thing that we are focusing here in pharmaceutical industry. We are making the drug formulations so that it meets the individual patient characteristics. Like for example, a particular person is allergic to a particular kind of drug, so we are making the personalized medicine so that the person can take the drug safely. Now, what are the emerging technologies? The new emerging technologies are like ambient mass spectrometry. We are performing the real-time analysis of their sample in their natural state. We don't have to extract out the sample. Say for example, if there is a sample in the plant, if there is a drug molecule is a bioconstituent in the plant leaf, we don't have to extract it out and check its mass. We can directly use the leaf. We are just doing the real-time analysis. 3D printing has been introduced in the drug manufacturing. We are performing customized drug delivery systems. And we are also using next generation DNA sequencing, obviously for the personalized medicines. Now, what are the implications of the pharmaceutical industry regarding pharmaceutical analysis? See, increased efficiency in drug development is very important, crucial, required. It is also required in drug development and manufacturing processes. We want to be more and more efficient. So we want to have a good analytical procedures to constantly check our workings. If we are having good analytical data, reliable analytical data, so it will help us to do the decision making. So a good, an accurate, a reliable data helps us to take the decisions in the drug development process. It will also helps in the improved patient outcomes because we are making the personalized and targeted therapies, so that will surely improve the patient outcomes. Now, summary for the chapter. So these case studies which we have understood, they illustrate the practical application. We have seen how they are being implied, how they are chosen, how they are implied, and how they are solving the problems. Real world applications, they are used for the quality control, formulation development, and the regulatory compliance. There are future trends that we have understood. There are future innovations that include advanced instruments, AI technique, personalized medicines. This is the direction where we are heading in our future. And obviously these advancements have very profound implication, very important application, because they will all improve the efficiency and efficacy of pharmaceutical analysis and obviously to our drug product. Thank you so much. So, dear students, this was all about our module. In the 10 chapters, you have understood what pharmaceutical analysis is and how it is applied in the industry. For an undergraduate student, these basic concepts are very crucial. You all must learn these concepts so that you can understand the bigger concepts. Actually, pharmaceutical analysis is a very large subject. So in order to understand the bigger subject, you have to understand the basic concepts first. So I recommend you to keep on practicing and please strengthen your concepts in the same, and we will meet you in the next module. Thank you.